The overall goal of this study is to purify outer membrane vesicles, or OMVs, from Legionella pneumophila in a standardized way and to analyze their influence on bacterial replication in human and murine macrophages.
This method can help to answer key questions in the field of outer membrane vesicle research. For example, a standardized procedure for OMV preparation may help to study their impact on host-pathogen interaction. The main advantage of this technique is that it will provide a standardized protocol to study the impact of Legionella pneumophila OMVs on human or murine macrophages. And this will improve our understanding of Legionella pneumophila pathogenicity.
We first had the idea for this method when we thought about the possible pro-inflammatory potential of OMVs and how this might influence the course of infection, as OMVs are released by Legionella both inside the host cell and extracellularly.
So the procedure will be demonstrated by Kerstin Hoffmann, a technician from my laboratory, and Lena Jung, a postdoctoral researcher.
To begin, while working under sterile conditions, spread L. pneumophila strain Corby on BCYE agar plates and incubate at 37 degrees Celsius for 3 days. Use the L. pneumophila from a pre-culture plate to inoculate 10 milliliters of YEB media at an OD 600 of 0.3 and incubate the bacteria at 37 degrees Celsius on a rotating shaker for 6 hours. Verify the purity of the liquid culture by spreading 100 microliters of the suspension on a blood agar plate. Incubate the blood agar plate overnight.